the science of biotechnology, which is a kind of a mixture of knowledge streams of microbiology, biochemistry, and other branches of life sciences put together, is what is biotechnology.
What's so exciting about biotechnology? Well, the decade that has passed has already been named as the decade of biology. Today, biotechnology is going to lead and it's going to be the forefront of every science. We are looking at Mars, we are looking at Moon. We have reached there, but how are we going to survive there? Well, it is the biotechnologists who are going to help us in that job.
Right from its applications in the area of agriculture for clonal propagation of high yielding varieties, the discipline of genetic manipulation of plants, from where you can fish out a gene from some species and add to the genome of some other species. Quite unrelated species have been used for this purpose. For example, brassica, canola is equivalent of brassica in certain countries. Genes for tolerance for frost have been isolated from fish and incorporated into the genome of canola. As a result, the crop has become resistant to frost damage. It can survive and grow well at very low temperatures.
We are talking about plant tissue culture technology, and as we all know, it means developing a large number of plants starting from a very small part of the plant. Now, what are the applications of plant tissue culture technology? We can develop clones. Clones means exact replica of the parent plant. So if you have an endangered plant and some very commercially important plant and you want the exact variety, so from a very small part of the plant you can generate a lot of amount of the plants, of course in vitro, that is in culture conditions or in the controlled environment. And what more, we can develop stress tolerant plants by genetic transformations, and these transformed tissues can further be generated in a lot of amount by tissue culture technology. And after you develop and grow the plants in laboratory conditions, they have to be shifted out in a natural environment. But before that, there comes the transplantation stage, which has to be carried out in the polyhouse. So we have again one controlled condition of polyhouse where things are a bit nearer to the natural environment, and we maintain it here for some time before the plants mature, and then they can be taken out. So we have a lot of plants from a very small part of the plant, and all these plants, all healthy, disease free, can always be propagated and shifted to the field.
What biotechnologists do? Biotechnologists modify living organisms so that they can survive better, they can be resistant to drought, they can be resistant to diseases. Everyone knows the name of BT cotton. What happened to it? It's a miracle crop. Just look at the farmers who have benefited from this BT cotton. Similarly, our crop has to be modified. We are looking at climate change. We are looking at various scenarios. We are looking at extreme climates everywhere.
In the form of forensics, in determination of the parentage, and in solving many crimes, because the imprint, what is now known as the fingerprint, is unique to an individual and a species.
DNA fingerprinting is a very important technique of modern biotechnology, where you can identify criminals, you can resolve the cases of the parentage disputes. Basically what happens technically, the genome of every human being, every individual has some codes, some genetic codes, hidden codes rather I should say, which can be explored to identify any criminal during the forensic investigations, and in the investigations where the parentage dispute is there and one need to confirm the biological parents of a particular child. In this method of DNA fingerprinting, several techniques of biotechnology are used, like gel electrophoresis. First, what we do in a flow chart, we isolate the genomic DNA from different individuals, then we do the polymerase chain reaction, we perform the PCR reaction to amplify those hidden codes, which are also called the molecular markers. And those molecular markers are then imaged using the gel documentation system. And these molecular markers or the DNA fingerprints, these have enormous variations from individual to individuals, and they are very much specific for a particular individual. So we can identify the criminals during forensic investigations just by identifying their molecular markers, and also we can find out the biological parents by looking at the molecular markers, because the progenies or the child share the similarity with the molecular markers of their biological parents.
So people are now thinking that it will be possible to do the gene therapy. Instead of the normal clinics, you will have clinics where the defective genes can be made fully functional, and diseases can be cured. You already know the case of Dolly, the sheep, which was a cloned sheep, and there are many examples where this type of cloning has been done.
You must have heard about bionic eyes, bionic limbs. All these are wonderful applications where engineers from electronics, mechanical, and all other sides, they integrate signals from our body with the help of biotechnologists and do wonderful things for the patients. Wonderful thing about biotechnology is entrepreneurship. Look at Biocon, the icon of India. It has become really one of the biggest companies of India, and it was just started by a person in the house just with very little sources. So that is the power of biotechnology.
And these days you are all familiar with the term IT, the IT sector. And sometimes people say IT is India's today and biotechnology or BT is Bharat's tomorrow. And all I want to say that this tomorrow is actually has already arrived, because biotechnology is now touching so many facets of our life.
Just imagine that a simple machine like BP monitor is not even made in India. Everything is imported. We are training our students so that they can be very good entrepreneurs, they could be very good engineers, and they contribute really well to the society.